Hi everyone, thanks for tuning into my talk today. My name is Denise Buckner. I'm a PhD student at the University of Florida, and today I'll be presenting some of my research on the assessment of electron beam irradiation as a lipid decontamination technique for light detection instruments. As an overview, first I'll discuss lipid biomarkers and their application for light detection. Then I'll introduce my group's novel light detection instrument, the extractor for chemical analysis of lipid biomarkers in regolith, or Excalibur. Then I'll discuss the importance of decontamination and introduce electron beam irradiation as a potential solution. Then I'll explain the experiments we conducted and their results. As a preview, we found that the technique was not effective at removing lipid contaminants of interest. Finally, I'll discuss my next steps and future research. In the search for extraterrestrial life, lipid biomarkers are organics of special interest. Lipids are universal to all life as we know it, primarily for building membranes that protect and segregate biological materials from the external environment. Cellular life requires encapsulation, and amphiphilic fatty acids are the organic molecules that fulfill this structural role. Lipids can form both biotically and abiotically, and have been detected throughout the solar system, particularly in carbonaceous meteorites delivered to Earth and Mars throughout both planets' histories. For reference, fatty acids make up over 60% of the soluble organics found in carbonaceous meteorites. Additionally, small simple hydrocarbons and chlorinated molecules have been detected on Mars by the Sample Analysis at Mars instrument on board the Curiosity rover, but further exploration is needed to better understand what lipids could exist on Mars and what the source of those lipids could be. Lipids bear origin diagnostic molecular features and patterns, meaning that the conformation of individual molecules and patterns and distributions of lipids within a sample can indicate whether those compounds were synthesized biotically or abiotically, a potential key to detecting extraterrestrial life. Lipids are also recalcitrant and robust molecules that can persist for billions of years in the terrestrial geologic record, which is orders of magnitude longer than other biomarkers like DNA, proteins, and amino acid and antiomeric excess. If life arose on Mars during its most habitable epochs about 3 billion years ago, lipid biomarkers could represent some of the only remaining molecular evidence of that life preserved there today. To search for lipid biomarkers on Mars and beyond, we are building the extractor for chemical analysis of lipid biomarkers in regolith, or Excalibur. This instrument will replicate and automate wet chemistry lipid extraction and analysis techniques that have been successfully used in terrestrial laboratories for over 70 years, streamlining and scaling them for in situ use on life detection missions. This image shows Excalibur's sample processing steps. First, Excalibur will accept a 50-gram regolith or ice sample, then comminute or crush the sample to reduce particle size. At this point, any volatile organics will be cold trapped and sent to an evolved gas analyzer. Back to the sample, lipids will then be extracted from the regolith via agitation in organic solvents, and the mineral residue filtered out, leaving behind a mixture of pure lipids suspended in solvent. The resulting lipid extract will then be concentrated 2,500 times to improve signal, then sent to a suite of coupled analytical instruments for detailed molecular characterization. These could include a gas chromatograph mass spectrometer, or GCMS, a thermocomolysis GCMS, or a laser desorption ionization mass spectrometer. However, if Excalibur is to effectively detect lipids, decontamination is essential. Lipid biomarkers are abundant on Earth, but may be low in abundance in an extraterrestrial sample. Since Excalibur will concentrate the lipid extract 2,500 times, any terrestrial lipids that end up in the aliquot will also become concentrated and sent to the analytical instruments. Those instruments coupling to Excalibur have ultra-low limits of detection with pico to femtomole sensitivities, so even trace amounts of contamination could present a significant problem. Further compounding the issue, recent studies on contamination in spacecraft assembly cleanrooms show that up to 10 to the 5th viable cells per square meter and up to 10 to the 8th total cells per square meter remain on hardware surfaces post-cleaning. This translates to roughly 500 picomoles of fatty acid per square meter. Finally, traditional decontamination techniques approved by NASA for planetary protection purposes, leveraged in cleanrooms, or applied in organic chemistry laboratories are either ineffective at sufficiently removing lipid contaminants or are effective at removing lipid contaminants but are incompatible with sensitive materials used to construct Excalibur and other life detection instruments. To address this issue, we investigated electron beam irradiation as a potential solution. Electron beam irradiation is a technique that's widely used for sterilization and decontamination in food, medical device, and wastewater treatment industries, and is sometimes used to sterilize spacecraft for planetary protection compliance, but has not been applied to life detection hardware in particular. Electron beam irradiation works by generating and focusing a stream of high-energy electrons onto materials undergoing sterilization. Decontamination is achieved in less than 60 seconds. The beam delivers tunable controlled doses that can penetrate up to 10 centimeters through cardboard, glass, and other packaging materials, potentially reaching trapped or encapsulated contaminants. These electrons can kill microbes and destroy some organics via radiolysis through direct hits to molecules and by generating radicals, but effects on molecular lipid contaminants had not previously been investigated. So the goal of our study was to test this technique as a final decontamination step that could be applied to the assembled Excalibur instrument. The intention was to enhance but not replace traditional cleaning. For example, the figure on the right shows how electron beam irradiation could factor into the Excalibur cleaning plan. First, heat-tolerant components would be baked to the highest allowable temperatures. Then throughout assembly, the instrument would be frequently flushed with solvents to remove any introduced contaminants. And these same solvents could also be used to pre-clean components that were unable to be baked. Finally, once the assembly of the instrument is complete, Excalibur could then undergo irradiation to destroy any residual lipids. So for this study, we first selected 5 target lipids to test, shown on the left, including a saturated and unsaturated fatty acid, an N-alkane, cholestanol, and cholestane. These molecules were selected for their astrobiological relevance and contamination potential. We took our selected lipids and deposited them individually into glass vials and sealed them each under high purity nitrogen. We then sent the lipid samples to undergo irradiation at Steritech sterilization facility at doses of either 0 kilograms, 50 kilograms, or 100 kilograms. Following irradiation, we used GCMS to analyze the lipids, quantify breakdown, and identify radiolytic products. We expected that the 5 lipids would be effectively degraded following treatment, but we actually observed no substantial reduction in the standards. This graph illustrates the abundance of each lipid relative to starting concentration after 500 kilograms and 100 kilograms of irradiation. You can see that the concentration is close to 100% for all 5 of the lipids at each dose, which indicates that no significant degradation occurred. The pink line, representing palmitic acid, shows that lipid abundance for this compound decreased to approximately 79% following 100 kilograms of irradiation, but no corresponding reduction was observed for the palmitic acid samples that were irradiated at 50 kilograms. Likewise, the green line, representing cholestanol, shows that the abundance for this compound decreased to approximately 84% following irradiation at 50 kilograms, but no corresponding reduction was observed for the cholestanol samples that were irradiated at 100 kilograms, so we conclude that none of our 5 standards are consistently or substantially degraded. However, while we observed no substantial reduction in overall lipid abundance, numerous minor breakdown products were identified in the irradiated samples, so some degradation or transformation is occurring. Radiolytic products include shorter or smaller molecules and recombinants. In these 2 example chromatograms from the saturated and unsaturated fatty acids that were irradiated at 100 kilograms each, we identified a homologous series of fatty acids and hydrocarbons of every chain length shorter than the parent compound. These represent breakdown products that were either decarboxylated or cleaved at various points in the hydrocarbon tail. We also identified recombinants, which include some of the molecules illustrated here, like the unsaturated fatty acid with 1 double bond that was formed after a saturated fatty acid with no double bonds was irradiated. We also see short dicarboxylic acids and oxoacids, which could represent fragments of 2 fatty acids that were cleaved and then combined together. We also observe a furanone, which appears to have formed via cyclization of the hydrocarbon tail around the carboxyl end. So what does this mean? According to our results, the 50 and 100 kilogram doses applied to the 5 lipid standards did not sufficiently destroy or remove the molecules under our experimental parameters, but some breakdown is apparent. Much higher doses could potentially be more effective at destroying lipid contaminants, but those doses could also damage or degrade instrument hardware, so we don't recommend using electron beam irradiation as a lipid decontamination technique for Excalibur or other lipid-based life detection instruments. However, the resilience of the lipids could suggest that these organics are a bit more resistant to radiation-induced degradation than we had initially expected. This is astrobiologically relevant because understanding lipid degradation rates in planetary environments where surface radiation is higher than on Earth is key for informing where and how deep to dig when searching for minimally altered lipids. The results are especially relevant to Europa, a body that experiences high levels of electron radiation sourced from Jupiter's magnetosphere. To follow up on this research, we're focusing on two sets of tasks. First, we're continuing to look for cleaning methods that can sufficiently remove lipid molecules without damaging Excalibur hardware. These include thermal, chemical, and laser-based methods. Second, we hope to further explore the effects of radiation on lipid lifetimes in support of future life detection missions to Mars and Europa. These analyses will include experiments that apply geologically relevant doses and types of radiation to analog samples made up of simulated Martian regolith or European ice spiked with lipids so that lifetimes could be quantified and breakdown products predicted. So finally, I'd like to thank NASA Ames Research Center for funding this project with the Center Innovation Funds grant, Blue Marble Space Institute of Science for supporting me during my research, the North Dakota Space Grant Consortium for providing me with a research fellowship, and the University of North Dakota's Department of Space Studies for facilitating my work. So thanks again, everybody, for listening to my talk, and at this point, I'd be happy to take any questions.